Hello everyone, and welcome to today's webinar. Population bottlenecks strongly affect the evolutionary dynamics of antibiotic persistence. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Bram van den Bergh, postdoctoral researcher, KU Leuven, FWO, VIB Center for Microbiology. Bram, you may now begin your presentation.
Hi, so good evening to you all, or good morning, or noon, whatever time zone you're in, and thanks for the nice introduction, Jennifer. And also, obviously, thanks to Inscripta and LabRoots for giving me the opportunity to share the work that we've done using the Inscripta technology. Today, I will be showing you that studying persistence, well, a type of resilience to antibiotics, that is important, and specifically that studying its evolutionary dynamics in the context of bottlenecks, population bottlenecks, makes sense to better understand our current battle against bacterial infections. In doing so, we discovered that the fitness landscape of persistence likely is rugged, and that these bottlenecks therefore can have strong impact on not only the course but also the outcome of evolution. So as Jennifer already told you, I'm a long-term postdoc in the lab of Jan Michiels, so the older person in the figure, leading our group at KU Leuven. I'm funded by FWO, but the talk of today is actually mostly spearheaded by Dr. Ethel Windels. It's a former student that I co-supervised and who recently defended her PhD, and actually she's not in the lab anymore, sadly. She went on to better future. And she's pursuing now a postdoc at the lab of Tanja Stadler at ETH in Basel. Obviously, we also got some support from other members of our lab, and also of Professor Tom Wenseliers, a professor at KU Leuven, an expert in biostatistics and computational biology. And of course, the reason that I'm giving the talk for you today, we got some support by Inscripta, who actually offered us the opportunity to work with their technology at an early stage. And the reason that we got into contact with Inscripta is through VIB. VIB is one of our sponsors, but it's also a research institute, and they actually offer us not only extra funding, but also this support TechWatch team that actually looks out for these new technologies. Now, let's go to the first intro, because I will give some intro on this topic. I think that's needed. The most important topic is for sure antibiotic persistence. This is the phenotype that we in our lab study on bacteria very often in many projects. And many of you probably think of antibiotic persistence as yes, we know this antibiotic resistance, we've heard of it, it's a big problem. And although resistance and persistence are very similar at many aspects, they do differ quite strongly at important aspects for this talk. So let's first go and define these things. So indeed, on the left, you can see the predictions of how many deaths we will suffer annually from failing antibiotic treatments, mostly because of antibiotic resistance. And this 10 million deaths in 2050, it's a prediction, so the numbers you can debate on them, but what is not debatable is the growing incidence of resistance. And indeed, this estimation of these numbers, this antibiotic crisis, is mostly caused by resistance. And what is resistance? That is shown on the right. With resistance, we mean that bacteria acquire the capacity to grow under elevated drug concentrations. So in green, you see a sensitive strain, and in red, you see a strain that has acquired resistance. It can grow at much higher levels of antibiotics, and a resistance level we quantify with this concentration at which growth can still be inhibited. The lowest concentration that can still inhibit growth is the minimum inhibitory concentration, or MIC. The MIC values go up as resistance increases. You also have commercial assays on the right slide. This strip is actually a gradient, or contains a gradient of antibiotics, and it produces a growth halo, and then the interface between the growth halo and the growth zone, that's then defined as MIC. It's a bit more easy to use that strip, for example, in a clinical setting. And measuring these MIC values is well established nowadays. Many people do it. It's quite easy to do. And obviously in a clinical setting, it's very easy to do as well, and therefore often done. And even we have these databases of MIC values, for example, the UCAS database, that contains a lot of MIC values of a lot of species towards a lot of antibiotics, which can be used then to decide whether a level is characterized as a resistance or not resistant level. Now, resistance is well studied, and most of the mechanisms are well known, I would say. It's mostly caused by mutations or by genetic elements that are transferred, and these genetic changes, they mostly impair drug target binding. And that will be later important if we compare it to tolerance and persistence. And since its link between mutations and phenotype is quite strong, this phenotype of resistance is therefore genetically inherited and passed on to the daughter cells, and very often also population-wide expressed. Now, in contrast to not being inhibited at a certain drug concentration, there's a second option of bacteria to evade drug action. And another option would be, especially if you think about bactericidal drugs, so drugs that kill bacteria, is to simply survive treatment. And that's what we call tolerance. And what would happen if you have a bacterium that cannot grow in high concentrations of antibiotics, but are simply not killed? Then this MIC value, if you would use the standard test that is used in clinical settings, this MIC value would actually not change. Why not? Because if it would change, then obviously we would speak of resistance. So the tolerance level can actually not be defined as the MIC, but as the killing rate. And that is shown on the bottom graph of the Browner reference figure, where you can follow the number of viable cells in time during a treatment. And if you then look at the susceptible strain or population, these numbers go down quite quickly. The more tolerant strain, although it is still killed, it's killed much more slowly. And there is even, so there have been some attempts, because this is a quite laborious thing to do, there have been some attempts to make it easier, especially for the clinical setting, to measure tolerance. For example, the group of Nathalie Balaban in Israel, they've come up with this minimal duration of killing, the MDK, to kill 99% of the population. But then again, it would still require a quite laborious setup to measure this MDK 99. Now, inherently, you cannot be tolerant to bacteriostatic antibiotics. So you need a bactericidal drug to speak of tolerance and also to speak of persistence, and I will come to that later. In contrast to resistance, this tolerance is also much more sensitive towards environmental conditions. So this killing rate can be changed quite strongly depending on the specific conditions in which you measure them. And basically, that's also one of the reasons, along with the laborious way of measuring tolerance, is that it's not or seldomly used or measured in clinical setting or looked after in during drug design. Now, The mechanisms, since it's also less studied in general, the mechanisms are less clear as for resistance, but most of the mechanisms or most of the results kind of indicate that some form of dormancy or inactivity is actually underlying this tolerance phenotype. Now, if we look, now that we defined tolerance, if you look at defining persistence, it becomes quite easy because persistence is actually a specific type of tolerance in a subset of cells. And we call these cells persisters. And as it is a specific type of tolerance, basically many of the aspects on the left actually also apply on the right. So again, there would be no change in MIC value. And to quantify persistence, you need to make those killing curves. You need to follow the viable cells in time during a drug treatment. Now, unlike tolerance, what you would see then is not a slower uniformic killing rate like you see on the left, but what you would see is biphasic killing, as you can see on the right. So initially you would kill most of your sensitive wild type cells, but then after a while in the persistent population or the population that has persisters, only persisters would be still alive and your killing will go down. Now, to quantify the persistence level, there are a few ways to do this. Mostly what is done is people use one time point of treatment and quantify the survival at that time. And as long as that time point is on the persister plateau, we would call it, so the second slower killing rate plateau, you're usually fine. The more correct way to do it would be to really determine those biphasic killing curves in full and fit them with an equation that is shown here. And this equation actually has two terms, the killing rate of normal cells and the killing rate of persister cells, so Kn and Kp. And from this equation, you can then estimate the number of persisters, which is in capital P, or the number of normal cells, which obviously you know. There are a lot of other ways to quantify persistence. You could even go as crazy and quantify the killing rate of the second killing phase. Or again, the group of Professor Balaban in Israel came up with the MDK 99.99, or the time, the duration of treatment to kill 99.99% of the population. But given that then you define your persisters as being lower than 0.01% of your population, I think you already bias your definition a bit. So I'm not really the biggest fan, and it's not popularly used anyway. Now, if we zoom into persistence, it's very important to realize that this is a form of phenotypic variation. So you really can think of it as a normal cell being switched to a persister cell, or a persister cell being switched back to a normal cell. And that this tolerance of this persister cell is only transient in time. Because of this, the link between the phenotype and the genotype is much, much weaker. So tolerance is genetically, the tolerant state of the persister cell is genetically not inherited to the daughter cells. And that will be important for the remainder of my talk. Unlike resistance, persisters actually have most, if not all, strains and species. Obviously, you have a lot of publication bias there, but lately people are seeing that there's a massive variation in the level of persister levels, in the amount of persisters present in each of the strains, even if you study them under the same conditions, because indeed, also, or similarly to tolerance, the environmental conditions are tremendously important to determine those switching rates or the persister level as such. Now, why these different isolates or strains or species have this kind of a variation, it's not completely clear, but the fact that all of them have at least some persisters has probably to do with the fact that it is believed to be, that persistence is believed to be a bet-hedging strategy. So some sort of insurance strategy against disasters that struck all of a sudden and that are quite important, that would kill your entire population. In contrast to resistance, it also protects against multiple drugs, high concentrations, sometimes even other stresses as well. And like tolerance, the mechanisms are very often not that clear. Many people detect different molecular mechanisms that are important. Most of them are related to dormancy or inactivity, as for tolerance, but I will not go into detail about that today. And people believe that they're important for the chronic nature of some infections. And we also showed, and I will come back to that later, that persistence can be a catalyst for resistance development. Now, if you then compare them head to head, and I will go over quickly. So in green, you have a population where you have a few resistant mutants. In yellow, you have a population where you have a few phenotypic variants, persisters. They would be killed the same way in a first treatment. All of the sensitive cells would be killed quite quickly. But then as soon as those sensitive cells are killed, the green cells, since they're resistant, they can start growing again, albeit slowly, while the yellow cells, the persister cells, don't do this. If you switch them to a fresh medium, then the green cells, obviously, they make up a population of composed entirely of resistant cells, while the yellow cells, they actually switch back to a normal cell state. They take a while sometimes, there's a lag phase sometimes, and then they actually make up a population that is, again, containing a few persisters and a lot of sensitive cells. So a second treatment would actually mean nothing for the green population. They're all resistant anyway, but would result in, again, a biphasic killing curve in the case of persistence. Then you could ask yourself, but why do you study evolutionary dynamics of persistence if the tolerance, the phenotypic tolerance, is not genetically determined? And that's a bit of a confusing thing, I must agree, but you have to realize that although on single cell level, the tolerance is not heritable, other aspects of persistence are genetically encoded. Most importantly, the number of persistent cells on the population level within this population, maybe also the lag time of persisters or the killing rate of those persisters, how they are depending on the environment. All those things are genetically encoded, at least in one way or another. So indeed, you can obtain mutants with an altered persistence level. And that's also why you can study persistence and persistence evolution, although, especially when I started my PhD, which is already a while ago, at that time it was quite heavily understudied, which is weird because there are a lot of science already at that time that showed that indeed persistence evolution is a thing, that it happens and that it's important. So what we saw already at that time was that different isolates actually showed widely different persistor levels, which is weird because there must be some evolutionary pressure causing this, especially if you assess them in the same test. Another strong indication that evolution is important for persistor levels is that late isolates from chronic infections that have been treated quite heavily with antibiotics is that they show often very high or higher persistor levels compared to those initial clones. And you also have those eco-evolutionary models on bet-hedging that predict that persistence would evolve, but also that there's an evolutionary stable state of the persistor level, and that's roughly that correlates roughly inversely with the good times in between treatments, if you think about it on an evolutionary scale. So basically, this was the start of my PhD, and I asked myself then a few questions. How does persistence evolve? How does persistence and its evolution, how does that affect resistance development, and which mechanisms of the plethora of mechanisms that we found in vitro, which mechanisms make sense in light of evolution? And you can already see that those questions are quite open-ended, not really the very focused ones you would write in a grant application, but in studying those things, we definitely found a lot of interesting things, and actually a lot of other students could continue on these questions as well. So I first came up with an evolutionary setup. On the left top, you can see this, where I would grow my E. coli cells overnight to stationary phase, treat them with a high dose of antibiotics, well above the MIC. In this case, we used aminoglycoside antibiotics. Those treatments would result only in persisters being alive. Those cultures would then be washed and diluted into fresh medium to repeat the cycle. And what we would see then is shown on the right graphs, a very quick and very strong evolution towards higher survival levels. So these data points are averages of 9 to 10 populations, and you can see that for the 4 antibiotics that I show here, we start off at a 0.1% survival or lower, and very quickly after 2-3 rounds, you actually end up with survival levels close to 100% even. Now, in this paper in Nature Microbiology in 2016, which basically started the evolutionary study of persistence in our lab, we've shown that this is not resistance, that indeed we still have. This is actually evolution of persistence. We also showed that this is very stationary phase specific, as we also designed our evolution experiment that way. And we saw costs attached to these high persistor levels, especially in drug-free conditions. Obviously, under treatment, they have a high benefit compared to the wild type, but in drug-free conditions, they have a pretty high cost. And we've also shown in that paper that basically the predictions by bet-hedging models are true, so that if you would treat less frequently than once daily, those curves would actually go down, would show slower and less extreme evolution. And we found that single mutations are causal to the phenotype. Now, as I said, this was a start to study evolution of persistence in our lab. We've repeated this trick in an AAC publication in other pathogens, basically, the ESKAPE pathogens, Salmonella, also uropathogenic strain of E. coli. All of them basically show the same kind of trends, where if you treat very frequently, you end up very fast with very high survival levels without any resistance. Now, brilliant minds think alike, or that's at least what I tend to believe or want to believe, because around the time that we were doing these experiments, other labs published similar approaches, most notably the Friedman paper in 2014 by the Balaban group in Israel. They have shown that if they treat daily or repeatedly upon dilution of a stationary phase culture in fresh medium, they would also get very high tolerance levels very quickly. Lucas Mechler's group has done similar work in the Bertram. Lucas Mechler in the Bertram group has done similar work with Staphylococcus aureus. And recently, in the last years, we've actually got a third modi operandi, where they didn't treat in stationary phase like we did, or they didn't treat upon dilution in fresh medium like the Friedman paper did, but they actually treated exactly in the middle of exponential phase. So basically now you have these 3 schools, let's say, of doing evolution experiments with persistence. But besides doing these frequent treatments and observing that you can obtain high levels, I also believe that in the last years, we've taken the evolutionary study of persistence to a level or a next level, which I categorize in 3 parts, let's say. So the most important one is that we've shown that tolerance and evolution of tolerance invites resistance development. And this was first shown again by Nathalie Balaban, where she showed, and this is shown on the figure on the right-hand side, if she continued her evolution experiment, that she would always first get these tolerance mutations in yellow or in blue, and then only afterwards she would develop resistance in her E. coli populations, always in a background of a more tolerant strain. And that's logic if you think about it, right? If you have a more tolerant strain, you have much more survivors, therefore the chance of you developing or of the bacteria to develop resistance is much higher. So this is a game of numbers. We showed, actually et al. Showed that if you have a highly tolerant strain or a more persistent strain, generally you also have an increased mutagenesis. So it's not only a game of numbers, but because of this higher mutagenesis, you also get in blue, you can see this on the right-hand graph, the OPPB strain, which is a highly persistent strain. You get a very quick development of resistance, much quicker than the wild type does in black, or than a lower persister strain does in green. And lately, the group of Dietrich Hart showed in his publication Nature that persistence can also form a pool of horizontal gene transfer that can happen more so with more tolerant strains. And it was also shown that tolerance can also catalyze the development of resistance to drug combinations, which is usually believed to be much more difficult. And then two other things that are likely to expand in the future is the study of epistatic interactions between mutations that affect resistance and persistence, or mutations that affect resistance only, and so on. One publication showed that already for resistance mutations and persistence mutations that combined increase tolerance much more than you would expect to based on their separate effects. And then very lately, we saw two large-scale in vivo observatory studies where they examined isolates of, in this case, Pseudomonas aeruginosa during fibrosis patients, and they basically confirmed what we've seen in vitro already on the evolution of persistence, that happens first. It catalyzes also resistance in these cases, so that's very nice to see. Now to the focus of this study. I know it took a while, but we wanted to know what bottlenecks would do on these evolutionary dynamics of persistence, and that was really Ethel's work. Now, why focus on bottlenecks, right? Why is that important? Well, to start, bottlenecks are inherently important, right? In evolution, because they basically are a balance, or they cause a balance between the force of natural selection and the strength of drift. Under very small populations that only have one cell, for example, there is no such thing as natural selection, and drift randomness is very strong, and basically all mutations can accumulate and fixate as long as they're not very, very lethal. Not only are they important in general evolution, but also they're quite important if you think about infections, because they're frequently encountered. So bacteria, they either transfer from one host to another, from one patient to another, and often this is involved in strong reductions in population size. But also, if you think about treatments, we impose strong bottlenecks on infections, on populations that cause infections because of treatments, and hopefully we completely eradicate them, but sometimes that's not the case. So therefore, bottlenecks can be important in evolution as well, and also because simply there's nothing known on how bottlenecks affect evolution of persistence. So that makes it inherently important, or at least interesting to study. While there's nothing known on specifically how it affects persistence evolution, there are obviously a lot of models and other studies on other traits that made us form a few hypotheses. What would happen under smaller bottlenecks? First, since smaller bottlenecks actually make you sample, or makes a population sample the fitness landscape suboptimally. I will show that later. We expect the evolution to lead to suboptimal outcomes. Since populations are smaller, or the bottlenecks are definitely smaller, we also expect a lower mutation supply, so a slower evolution and perhaps a longer waiting time. Since the effective population size is smaller, we also thought that the few mutations that will occur, that they will actually fix way quicker, because it's only a smaller population that they have to fix in, let's say. So we expect a lower intra-population diversity. So the diversity within a population, we believe should be lower. And if you look at different populations, since populations will randomly sample the genotype space, each of those populations will do that, we expect a higher interpopulation diversity. But most of these hypotheses are highly dependent on the topography of the fitness landscape. And with that, I mean these two graphs that I showed you here. The fitness landscape on top actually shows in the plane all the possible genotypes, a reduction, obviously, of all of the possible genotypes. And then the height of the peaks shows the fitness, and in this case, also the tolerance levels. If you have, in this very simple and weird, simplistic fitness landscape, if you only have one peak and then it doesn't really matter whether you have large populations or small populations. Each population will eventually evolve towards this one high peak, high fitness peak. Now, if you have the situation in the bottom, which is much more complex, you have a lot of suboptimal peaks at different locations, you also have valleys, then the size of your population matters a lot. So the red population actually contains a lot of different genotypes at the start, and therefore all of the replica populations will likely evolve towards this peak in the middle that is also the highest peak. Now, if you take a smaller population, then it highly depends on which population you take. By accident, you can start with a yellow population that is more closely to the peak on the left top. Or you can start with a blue population, and that is more to the bottom. As a result, those two populations, they will evolve differently, and they will end up potentially at different suboptimal peaks. So those were the hypotheses to start with. Now, to examine this, Ethel had to adjust my setup slightly, so that she could do way more replica populations. So instead of large volumes in flasks, she used microtiter plates, and she used the same cycle, so a growth cycle of 19 hours, and then a treatment of 5 hours. And in between, obviously, she washed to remove the antibiotic, but she also imposed bottlenecks. So taking only a certain fraction of the treated volume to inoculate the next cycle of between a 1 to 500 and a 1 to 10 dilution. And in doing this evolution experiment, she did 40 populations per bottleneck conditions. She obtained, well, the evolution that you can see on the right-hand side, where the red curve, the 1 to 10 dilution, resembled quite strongly the outcome that I had in the larger volumes, a very quick and very strong evolution. And the smaller the bottleneck becomes, the slower, the more extreme, and also the longer we had to wait for evolution to start off. And to quantify these three parameters, so the speed, the extent, and the waiting time, we fitted these data with a model that describes basically the spread of a single mutant in a population. And this model that you can see there below the graph, it contains a few terms that are interesting. So the P is the persister fraction, and you can see there an S. This is basically the slope or speed of evolution of the graph, of the sigmoidal graph. And then the lambda factor is basically the lag time or the waiting time that the population needed for a mutation to occur. So I forgot to say, all these points that you show are obviously averages of many evolution experiments that I don't show, but that you can look up in the MBE paper of Ethel. So if she did this, so yes, indeed, that's what we expect, those three errors, or what we see. If she looked at those three parameters, then you can indeed see that bottlenecking restrains evolution. So not only is the persister fraction, the end persister fraction, lower at smaller population bottlenecks, but also is the rate or the speed of adaptation lower at lower bottlenecks, and is the lag of adaptation larger at lower bottlenecks. So all these things significantly correlate the way we expect them to under our hypotheses. Now, obviously, fitness is the parameter that evolution selects upon. So to speak of restraining evolution and to speak of rugged fitness landscapes and so on, you actually need to verify that indeed fitness is also agreeing with these hypotheses. So what we did to measure fitness rather than survival, which likely is a very good proxy for fitness, but nevertheless, we performed head-to-head competition experiments where we mixed an ancestor strain with a population that was evolved. And you can generally see in this left graph that all of the populations have an increased fitness, which is logic, right? Because under our cycle of treatment and 19 hours growth, they were all evolved on, they all adapted towards that condition. So it would be strange if they are not better at growing under those conditions than the wild type. But there's a nice correlation again between the bottleneck and the relative fitness. So the highest fitness is for the bottleneck that is the largest. Obviously, this fitness can be made up of the survival that we already measured, but it could also be that, well, you have this 19 hours of growth in our cycle, that something changed in that time period as well. So we measured growth in a semi-automated way, measuring OD at regular time intervals for all of our bottleneck groups. Obviously, I only show you 2 groups. And what you can already see is that while the ancestor, it has a dioxic shift here in the middle, but for the rest, it nicely grows over that time period. The evolved mutant actually shows worse growth, right? It has, especially after the dioxic shift, there's some impairment of growth. And if we then do this for all of the conditions and measure the AUC, being the area under the curve, we can see again that there is a correlation between the bottleneck size and the area under the curve, but a negative correlation, meaning that the costs of fitness, which we already knew is usually present under drug-free conditions in high persister strains selected in these evolution experiments, that the cost is higher the better the population is adapted, because indeed the 1/10 population is the one that has the highest fitness and also the highest persister level. So the largest cost in the larger, better adapted bottleneck condition. Obviously, this was all done on a phenotypic level, so we switched to a genetic level because, again, our hypotheses were actually formulated on the genetic level, on this fitness landscape and the genotype space. So what we did, we sequenced all our populations, really the populations as such, at a deep level, deep coverage. We determined frequencies of the mutations, and when we aggregated our data on the left, we could already see that the distribution within the populations was quite different depending on the bottleneck size. So the small bottleneck populations, they actually had a few very high frequency mutations at the right. They had no intermediate frequency mutations, almost none, and they had a very high level of low frequency mutations on the left. While if you increase the bottleneck, those intermediate mutants, they do occur much more popularly, actually, at a quite high amount, because these are the distributions of those frequencies among all the replica populations, meaning that likely these populations are so large that you can have interference of different mutants trying to fix but never actually succeeding in doing so. And well, if we look at the specific targets, we see a similar thing. So the left-hand side basically shows for a low intrapopulational diversity. So within a population, there's not too much diversity. It's either in the small bottleneck condition, that is, it's either fixed or not fixed at all, nothing in between. But if we looked at the target specifically, then overall we could see that one specific target dominated our data set, which is the nuo-operon, encoding for complex 1. I will not come back to the mechanism or the expected mechanism. We have been studying this mechanism, or this operon, actually for a while now, but this was a very popular target. But if we then looked at how popular it was obvious that this was a less popular target in the small bottleneck condition, where we obtained much more other highest frequency variant mutations. So in blue, you can see all the nuo mutations, and in gray, you can see all the other mutations, or at least the genes in which they occurred. And the way we quantified this is basically we counted the populations. For each population, we determined the most popular or the highest frequency variant, and obviously the target where the mutation occurred in. And then for all of the replica populations, we basically compared them and counted them. So here you can see that there's much more interpopulation diversity at lower or smaller bottlenecks. But you already feel that this kind of a setup is actually not well controlled. There are a lot of reasons why this is not an ideal setup. For example, there are tons of possible mutations that can occur. So you might even need more than 40 replica populations to really make this make strong conclusions from this. You're very dependent also on the sequencing quality and quantity to detect very low frequency mutations. We only based ourselves here on SNPs, not on larger rearrangements. So all these things made us believe that we needed a much more controlled condition. Yeah, indeed, there's a correlation there, although it's not really significant. But this much more controlled condition that I'm talking of, luckily, at that point when we were looking for such a condition, we were brought in contact by the VIB, by its TechWatch team, with Inscripta, with this emerging company that was looking for applications for its technology that is basically still roughly the same as its original publication. What did they do there? They target a certain region in the chromosome with this spacer, with this guide RNA, with CRISPR-Cas, and at the same time they link this spacer to an homologous arm that at the same time then mutates this region to a mutation of choice. Obviously they upscaled this to a highly multiplexed setup where they used computer-aided designs and high throughput synthesis of oligos, so that they can do multiplexed recombinering in a pooled format, creating many different mutations at the same time. And this guide RNAs and this cassette, basically, it was provided on a plasmid. So basically, those mutants, they still contain a plasmid with a guide RNA that can act as a barcode to detect the frequency of mutations in a selective enrichment condition, where then before and after the selection, you can basically measure the frequency for each mutation in a simple way by just extracting the plasmid and doing deep NGS barcode profiling. And then by comparing the frequencies before and after treatment, you can get an estimate of the fitness of this mutant under those conditions. So Inscripta delivered us two types of libraries, a KO library in E. coli containing 4,000 and something knockout mutants in a pooled fashion. This resembles a bit commercially available arrayed libraries, but this is obviously much more practical to us since it's pooled and we can assess it also in a pooled fashion. We don't have to screen anything. And then a second library, the non-coding RNA library, a library with only 87 knockouts that remove or delete a non-coding RNA gene, for which most of them actually are not present in literature or definitely not in a commercial format. We applied 2 rounds of our selective regime with 6 different bottlenecks, ranging from transferring 1 to 5,000 to 1 to 10 of the population, and we did 8 replicas for each of them. And obviously we profiled the barcodes with NGS and applied count-based statistics in the analysis. And when we do so, we actually first looked at what percentage of the mutants we maintained. And what you can see in the top, but also in the bottom for both libraries, we see that the smaller bottlenecks, or the smaller the bottleneck is, the more of the library you lose, which is logic in a way. But this already indicates that you get, by applying small bottlenecks, you get a stronger decrease of the intra-population diversity. And again, these things were significantly correlated. But also, if you look at the frequency distribution of the mutants per population, you see the similar thing as we saw before, namely that maintained at a very strong small bottleneck were predominantly made up of mutants at a very low frequency, and the higher or the larger your bottleneck becomes, the more this distribution shifts towards intermediate frequencies. Now, the nice thing here, obviously, is that you have these barcodes, and you have counts for these barcodes for each of the replica population in each of the bottleneck conditions. And therefore, you can make correlations between each population and each barcode within that population and all of the other replica populations of that condition. So that's what I show here for one condition. So you get those triangle graphs. Obviously, in the top graph, you get a lot of dots because we have a lot of barcodes there, a lot of mutants. The bottom, the ncRNA library, is much more less complex. But you can do that. You can have actually a correlation measure for each of those sub figures or subgraphs, convert them to a color scale, and even compute an average, which is in the top left, an average correlation value. And you can see that correlations between replica populations are significantly weaker at the smaller bottlenecks. Actually, the smallest bottlenecks for the ncRNA libraries died off, but that's nevertheless not a problem because that same kind of trend is visible for the ones that did survive, which means that you basically have a higher interpopulation diversity with smaller bottlenecks. And also, if you look at the specific targets, the same thing is true, right? As we showed before. So for the KO library and the ncRNA library, we had two very popular hits at the large bottlenecks, the 1/10, namely the delta CRR and the delta MICC. But this popular target was less popular in the populations with a smaller bottleneck. And again, this is significantly correlating. The funny thing, though, was that in the KO libraries, we only could significantly detect enrichments, likely because of the complexity of this library. Depletions could be depleted for all sorts of stochastical reasons. And with the ncRNA library, we could only find depletions, potentially because deleting an ncRNA is very costly in general. Now, let me conclude. So small bottlenecks, we showed that they impair persistence evolution because they have a slower adaptive rate. They end up at lower end levels, but they also increase population diversity and decrease intra-population diversity, which both matches with the rugged persistence landscape. And we also detected novel genes or mutations linked to persistence. This is shown on the graph at the right as well, but because of time, I will leave that to you to look at a later stage or to look it up in the publication. And for the future, well, obviously we want to continue on this study of the evolution of persistence, see whenever selection leads to resistance or persistence, how this happens, why this happens, when this happens. But also look at the diversity, the genetic diversity that remains hidden, we believe, in a large-scale setup by the very popular targets that we initially pick up there. And I'm also looking specifically at whether there's such thing as a cost-free persistence or whether the cost of persistence is very inherently linked to persistence. Now, for the Onyx platform, the reason that you guys are probably here, the ideas are less, I would say. So we are working with the Onyx platform that we recently obtained to make gene saturation libraries, constructing all possible mutations within one gene on the genomic level to see which alleles can affect persistence, but also other traits, obviously. But we will definitely also construct more complex combinatorial mutants, and like I said, the options are limitless here. And with that, I would like to take any questions that might have arisen. So I give the word back to Jennifer, I think.
All right. Well, thank you, Bram, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the relevance of less optimal peaks? Are we not simply interested in the most optimal peaks?
Yeah, so naively indeed, I think it's a very good question, but naively you could say, why do we want those suboptimal peaks, right? I mean, the ones that make this phenotype the worst, let's say, in a clinical setting, those are the ones that we want to tackle. But that's not always true. I mean, these are very artificial conditions still. This is a lab environment that we select our mutants under. So it could be that some of the mechanisms or some of the mutants that we select are not necessarily very popular in vivo or not very relevant in vivo. To so if we can detect the entire tolerome or persistome, or how do you want to call it, all the mutations that can affect persistence in one way or another, it can obviously lead to a better understanding, and therefore a more complete picture, let's say, of the phenotype. So.
All right, well, thank you very much, Bram. The next question, is there a connection between PPGPP and persistence? And he says, also great to see you. Maybe someone you know.
Yes. Yeah, David. It has been a long day indeed, David Marciano. Nice for the question. Thanks for the question. So, is there a link between PPGPP and persistence? Yes. So many people have found this link, have studied this link. Mostly related to toxins, sadly, and this story has been retracted recently, but still PPGPP, the alarmone, so the stringent response alarmone, has been involved. And generally what we see is if you remove PPGPP by deleting Raleigh and SPOT, generally what everyone sees is that you have fewer persisters. I must say though, in the mutants that we generally select in these evolution experiments, we don't see these things. We do not see a dependence on PPGPP, which is a bit weird because PPGPP is also involved in the stationary phase response. So upon entry in stationary phase, where we also see an increase in persistence and a specificity in our evolution mutants towards this stage, there is an increase in PPGPP, but if we remove it, it doesn't really seem to matter. So is this specific to our outcome? Perhaps. Is it specific to our conditions? Very likely. But in general terms, people have this. Have studied PPGPP dependent persistence in the past. Yes.
All right. Thank you so much. Our next question: would you predict that the type of bottleneck, e.g., dilution versus antibiotic, determines the types of mutations that occur?
When I first read the question, I was thinking no. So my initial answer would be no, because the bottleneck is the bottleneck, and I think the way you obtain the bottleneck does not really matter in terms of how the heterogeneity is affected and how the speed is affected, but the types of mutations that you select could be still depending on whether you treat strongly or less strongly, or whether you just dilute. Obviously, if you just dilute and you do not treat at all, there's no selection for persistence mutations. So then definitely you would get other mutations. But if you would still combine the two, but play around with the strength of both, let's say, I think it might matter, because if your treatment is less severe or much more severe, obviously it could be that some mutations are specific for the severity of this treatment. Yeah.
All right, thank you, Bram. Next question: why did you use aminoglycoside antibiotics? Yeah,
so the aminoglycosides, they're one category of drugs that we used. We used that as a start, and that's one of the reasons why we also continue with it. But the better reason to use aminoglycosides is that it's a bactericidal drug to start with, and that it is quite efficient to kill stationary phase cells. And we really wanted to work in stationary phase for several reasons, and I can go into that later for several reasons. But that was one of the reasons that we had to. Well, our choices were quite limited, to be honest. Fluoroquinolones would be another option. We've not tried that yet in full scale, let's say. But aminoglycosides were also the first where basically this evolution was quite reproducible. So that was one of the reasons as well.
All right, thank you, Bram. Next question. You have shown a graph that shows disruptive selection in antibiotic persistence. Would this mean that even though the microorganisms are of similar species, the drugs to kill these microbes will differ, like a different drug or for another extreme?
So I believe what the person who asked this question means is that I have showed that evolution also works for other species, but that indeed depending on the species, but also depending on the drug that we use, the absolute starting level is a bit different. Maybe also the absolute end level might be a bit different. The reason why we chose different aminoglycosides is just to make our conclusions more generally valid, let's say. But there we do see even different end levels. So, for example, gentamicin, we see that in the lab as a very, very potent aminoglycoside, resulting in very low levels at the start, but also lower levels at the end. And for the pathogens, there we chose the antibiotic that at the start resulted in at least a biphasic killing pattern. And for example, for Pseudomonas, we used, I think, gentamicin, and Pseudomonas aeruginosa started at a very, very low level with this type of treatment. If we would use other drugs and other concentrations, this starting level might differ a bit, and maybe the end level as well. But I think by now, because of all the other groups that also use evolution experiments, the conclusions would largely remain the same for those kind of experiments.
It looks like we have one last question here, Bram. Why did you use stationary phase for the evolution experiments?
Yeah, so we, at a given point, I had to choose anyway, but we used stationary phase in the design of this evolution experiment. But as I showed you, many others used other setups, made other choices. The reason that we used stationary phase is both practical but also biological. Practical because stationary phase, if you from one day to another vary a bit, you're still in stationary phase. And although I do realize that early stationary phase or 20 hours later, that can still matter a lot, but if you are still changing 1 or 2 hours in your setup, it's probably not the biggest deal. While if you do that in exponential phase or upon dilution in fresh medium, like the other groups do, that can change a lot. Another practical reason that we chose stationary phase is that if we treat in exponential phase, well, we kill almost all of our cells with these kind of treatments. And I was already worried in stationary phase that we would be on the edge of what would be viable, because after a while, I do not want to start with 100 populations in flasks and then after a while only have 1 or 2 viable populations left. So actually that was one of the reasons to work in stationary phase, because the levels are generally higher there anyway, but also because was also one of the reasons why I worked in those large volumes. Then later et al. Proved that was actually not so important, sadly. So I did a lot of work for nothing. But then the biology behind it, I also tend to believe that if these are very artificial, I realize, right? But if we continue to study bacteria in mid-exponential phase, then probably the relevance to in vivo conditions where mid-exponential phase, I don't believe that there is such a thing as mid-exponential phase. I'm not saying that they're all in stationary phase in vivo, for sure not. But to me, this sounds a bit or resembles a bit better what might be happening in vivo, where conditions are far from optimal to grow exponentially. So that's a biological reason to use the stationary phase, in fact.
All right. Well, thank you. It looks like we don't have any more questions. Thank you, Bram. Do you have any final comments for our audience today? No. Just that we are very happy with the Inscripta platform, and that's obviously why we acquired the Onyx commercial machine now as well. And I'm very anxious to see it in real life. I didn't yet because of Corona measures, but I will in the near future. So I'm looking forward to it. Well, thank you again, Bram van den Bergh, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.